Welcome everybody for this GHGA lecture series on advances in data-driven biomedicine. We have the honor to have today Ivo Gut from the Centro Nacional de Análisis Genómico, which is in Barcelona, one of the largest genome sequencing center in Europe. So Ivo has worked before at Ivry, near Paris, where he was at the Centre National de Genotypage, where we established the highest throughput genotyping platform in Europe, and has since then also endorsed many responsibilities in the field. He's been a principal investigator, for instance, in Beyond 1 Million Genomes. He has been coordinator of the RD Connect, and is part of the international Rare Disease Research Consortium. So he's active in many consortia, including the ICGC in cancer, the International Human Genome Consortium, and the International Rare Disease Research Consortium. Ivo, we are very happy to have you today. I want already to announce you the upcoming talk. So this will be on the 13th of December. Helen Parkinson from EBI will give a talk on data integration strategies for biomedicine. A few points now regarding this lecture, a few organizational points. All participants are muted, and there will be a session of question and answer at the end. So if you have questions, please ask them through the Zoom chat function anytime. I'll go through them at the end. And note that this lecture will be recorded, and the link will be provided on the GHGA webpage. Ivo, thanks a lot for giving the talk today. And without further ado, the stage is yours.
Okay, thank you, Julien. Yes, okay, I first have to try to share my screen. That's what I'm going to do now, see if this works. Share. Put this to full screen. Okay. I think this probably should be okay now, and I hope that you can hear me fine. Good. I want to thank Julien and the German Human Genome-Phenome Archive for the opportunity to come and talk to you today. It's a great pleasure, and I will try to give you more or less a very quick tour of what we've been doing in infrastructure projects. So, although Julien actually asked me in the context of presenting Easy Genomics, which is an infrastructure project that I coordinate, and I noticed that I'm actually only touching on that once. What I'm going to mainly talk about is even larger infrastructure projects. Okay, start off with, I want to talk about the 1+ million genomes. It was mentioned before already. The 1+ million genomes is not a project. The 1+ million genomes is an agreement of different countries signed usually by health ministries to make genome information that is collected in the context of clinical diagnostics available across country borders, and obviously to do that in a secure and equitable way. At the moment, there are 24 countries that have signed this. 23 of them are EU countries. There's Norway on top of it. And then there are 4 observer countries. And as I said, the objective is to share 1 million genomes, high quality genomes across borders by the end of 2022. Now I will tell you that we will not completely get there until in 1 year's time, but we're moving in a very good way. So, as I said, 1+ million genomes is not a project. It is just it is just an agreement to do this, but there's no money behind the 1+ million genomes, which is why the European Commission put up a coordination action, which is called Beyond 1 Million Genomes, that then helps to actually move forward into this objective of the 1+ million genomes. It's a project, it's coordinated by Elixir, Serena Scollin, and it is funded with 4 million euros. And it works all of the design, the testing, the putting it together, the moving it, all of the elements that you actually need to be able to do this. I'd say sort of off the bat here, it is a lot harder than we would have imagined, but we've made a lot of progress towards this, and there's many different screws and things that we have to work on and tighten and get together and coordinate and so on. And then obviously the idea is beyond this project to then make this sustainable and move beyond the 1 million genomes that we want to do now. So the 1 million genomes is sort of in a way just the test bed, the proof of concept. This is then meant to go into full implementation in the future for the benefit of the European citizens. So many different working groups, many different parts that are playing together with this. What you can see here is obviously you want to do this under legal and ethical conditions that are acceptable. We want to define standards that you can do this along. We need a federated, secure cross-border technical infrastructure. You want to deploy this in the context of personalized medicine, and you have to make it sustainable. You want to involve industry because they have a major part in this, and you want to have a set of use cases for which genomic analysis is actually of benefit. Rare diseases is an obvious one. Cancer is another obvious one. Common complex disease is important in terms of predicting who might go in which direction or might come down with which disease in function of their susceptibility. And then, as we all know, a couple of other surprises came in the last couple of years, such as COVID-19, which then led to the establishment of a use case group working around COVID-19. And then very recently, an initiative to put together a European reference genome. So essentially sequence on the order of about 500,000 genomes of healthy individuals and put them at the service of the population. For example, in the context of common complex diseases. So do this predictably large scale. There are many stakeholders in this space. Obviously, the citizen stands at the center of all of this. There are partner projects, a lot of EU projects that are doing bits and pieces that are of big relevance for the beyond 1 million genomes. We work together with them, and we talk to them from time to time in stakeholder forums. So the first one I'm going to talk about is this standards and quality guidelines. I chair what is called Working Group 4, which is a working group called Good Genomic Practice. In the context of the beyond 1 million genomes, this working group is joined together with the working group that is responsible for defining the ontologies. Just quickly, something about the ontologies. One of the really important things is what you actually call the diseases that you are looking at, and how do you put them together with the genomes that you're sequencing. And there are many different standards that you can use. For example, in cancer, very popular is this SNOMED-CT, particularly used by pathologists, but healthcare systems in general. So how do you do this? How can you sort of share information on the phenotypic or clinical descriptions of the patient? So this working group works a lot in this direction. Then, as I said, work package 3, working group 4, which is the working group that I chair, with the title Good Genomic Practice. And inside this, we are meant to define guidelines for how to do genomic sequencing, just the technicalities of whole genome sequencing and exome sequencing. Now, how did I get the pleasure and honor to chair this group? Well, the reason is that in the International Cancer Genome Consortium that ran from about 2008 to about 2019, 2020, That was an international effort to distribute the genomic analysis of different types of cancer across the planet, so that not every nation does breast cancer, prostate cancer, lung cancer, and colon cancer, which are the four most common cancers in any country on the planet, so that essentially all forms of cancer would be worked on in parallel, distributed across the planet. And so that project, over the 10-11 years that it ran, sequenced on the order of about 25,000 cancer genomes. And essentially the idea was to put together somatic profiles of the patients into a common database. So do the same experiment, sample a tumoral tissue, sample a comparative normal tissue from the same individual, do the same analysis on both, for example, exome sequencing or whole genome sequencing. If you can afford it, do DNA methylation analysis on top of it. If you can afford it and technically are able, put RNA sequencing on top of this as well. So in principle, a very simple thing. Take a sample of one, sample of the normal of the same patient, do the same analysis, compare the two, identify the differences. So the somatic mutations that are present in the tumor but not present in the normal, and then stick them in a database and do this for each cancer entity roughly 500 times. So do it for 500 patients. Now, very early on in the project, Tom Hudson, who chaired the ICGC, talked to me. He came up to me, I think it was even at a meeting in Heidelberg, and he said, you know, everyone is doing this genome sequencing. Do you think they're doing it the same way? And I said, well, I'm absolutely certain that they don't do it the same way. And he said, well, what should we do about that? I said, well, the best thing is, we just compare everyone against each other and try to figure out what they do. And so I was put in charge of a working group there that was meant to figure out how people do their sequencing. Now, sequencing is not a straightforward garbage in, garbage out project sort of problem, but a rather more complex process. If you key it up very crudely, you have the data generation and you have the data analysis. And obviously, only if you get good data and good analysis do you get results that you actually can rely on. So I was put in charge of figuring this out. So the thing we did is, together with Data-Driven Ivo Gut, we took a patient and we had enough tumor material and enough normal material from this patient, and we distributed to whoever wanted to sequence this. There were 5 big sequencing centers that put their hand up. CENAC was one, Data-Driven obviously, OICR, RIKEN, and Sanger. So we all got the same material and we all did sequencing on the same materials. Blue is the normal of the patient and yellow is the tumor of the patient. What is a good result? A good result is, so this is normalized TC coverage. Good is when it's horizontal. Horizontal is a good result. Everything that deviates from horizontal is less good. The tumor has a duplication of a couple of chromosomes, so it splits up into multiple levels. And because the patient is male, there is this line down here. Okay, but what you can see is even big sequencing centers doing the same thing looks very different. So for example, this one here looks very different than this one here. This being a good result, this being quite a bad result. Or in some instances, you have situations where one looks reasonable, so tumor looks reasonable, but the normal looks bad. E, C, D, and E are the 5 big sequencing centers that I mentioned, and Data-Driven, when they saw this, they said, oh, we'll try all other protocols that we can get, or vendor protocols that we can get our fingers on, and they went and did that, which is F, G, and H. And so the story is, we were doing things very differently, even though we had exactly the same machines, the same materials, the results do not look the same. So people sequencing do not necessarily produce the same results. Next thing we did is we said, okay, let's go at the second part, the data analysis part. And so what we did is we gave one of those data sets, the one of A that was fairly good, two bioinformatics groups and said, analyze them, tell us how many somatic single base mutations there are in this sample. And this is what happened. So we had 18 different groups contributing, and what we saw was this. There are roughly 1,200 somatic single base mutations in this tumor. We generated a gold set by taking all of the data that everyone had sequenced, putting it together and running it through multiple pipelines and consolidating this. And so we knew that there are roughly 1,200 somatic mutations of this type in this particular tumor. And then you have all the 18 submissions. Now, with the same data, exactly the same data, everyone could agree on roughly 200 of the somatic mutations that actually are there. So that's what is in red. And then everyone -1 is in blue, everyone -2 is in green, and so on. So you see, even having exactly the same data, there's quite some disagreement when you just look at substitution type mutations. When you look at somatic insertion deletion mutations, the story is even more bleak. There's roughly 350 or so in this genome, and you see that out of 16 different analytical teams doing this, everyone can agree on exactly one somatic insertion deletion mutation. Then everyone -1 is in blue, everyone -2 in green, everyone -3 in this purple here. So a lot of disagreement when it comes to identifying somatic insertion deletion mutations. So this is rather bad news. So, you know, you do the same thing, you think you have the easy part, and the results end up very differently. So you can obviously, once you've done this, you know what the ground truth is. You can line everyone up, and you can define a precision and recall. So you can identify groups that are very, very precise, but they don't find everything that's there. They only find 40% of what's there. You can find ones that are reasonably good. They find sort of a reasonable number of the somatic mutations. One would be the perfect call here. And then you have people who call a lot of stuff, but 80% of what they call, or 90%, is actually wrong. So this is sort of the landscape when you look at substitution mutations. And when you go over to somatic insertion deletion mutations, it's even more bleak. No one gets anywhere near this 1 point here. So very difficult to do. With this data set, you can also try to figure out where and why did people actually go wrong. Well, here we've just lined up the somatic mutation calls, colored them as true positive. So blue is correct. False negative, so this was missed, and false positive, this was a call that someone made. This is actually false. So the first example here is good. This analytical group is quite competent. The second group has a problem always in defined positions in the genome. It turns out that this here is as soon as the sequences are relatively close to the centromere, the caller basically falls over and makes calls that are not actually there. This caller here is just bad across the board, calls far too many things. It does not really do a good job. And then this one here has a problem in the chromosomes that are duplicated. So if there's an imbalance between the normal and the coverage of the tumor, or the copy number of the tumor, the somatic mutation calls go off and they start producing false positives. So this was in at the end of 2016, we published this. People look to this piece of work very often and use this set to sort of benchmark their pipelines and their work. So now, was that a representative test? For the time, it definitely was a very representative test. It still is good enough. However, there's obviously, technologies have moved on. We know how to sequence better. We know how to key things up better, and so on. So good genomic practice, the working group 4 of the 1+ million genomes, sort of was put in to continue on this problem. And essentially, what we said we would do is we would put up comparisons. So we would put up studies looking at tumor normal comparisons by whole genome sequencing, whole genome sequencing applied to germline variant calling, and then the same for exome sequencing. So this we did with beyond 1 million genomes and with Easy Genomics that has the standardization work package. So we're putting that in here. The objective of all of this is to figure out how good people are doing their sequencing and how good they are doing their data analysis, and to provide guidelines for good genomic practice for the specific application. So what's going on at the moment? So we're just about at the beginning of kicking off both of these benchmarks. I should say, kicking off a benchmark and kicking off an interlaboratory comparison. If someone wants to know more, I can explain the difference between the two things. It's taken a lot of preparation. You have to go through. We want to do this on the same type of materials that the normal analysis will be done on. So it has to be a representative sample. Due to COVID, procuring the tumor DNA samples has been very, very complicated, and the germline, we had to go through ethical approval for this, which also was quite taxing, but we have that now. So essentially, I'll try to explain a couple of things that go with this. So the first is we have the germline. This is periodic interlaboratory comparison, and might be underneath all of the bumps that comes from Zoom there. So what we're doing here is we are providing a germline sample. We are expecting people to do whole genome sequencing using Illumina, produce FastQs and SAF files, give those to us. We will also use the FastQs to merge all of the submissions and do variant calling out of that. We have on top of the proband sample, we have samples from the parents. That means we can test the genotypes. We will run all this through the analysis pipeline. On top of it, we will do an orthogonal technique of nanopore sequencing, and then build out of that a gold set of variant calls. All of the participants are meant to provide variant calls as well, and then we can compare the variant calls that are submitted with the variant calls of the gold set and say, you are so and so good. And this will give a performance report for each of the laboratories that's participating. For the somatic mutation benchmark, it looks similar, except there's going to be 4, maybe 5 tumor normal pairs. Again, sequencing should be done with Illumina. There's one entity that signed up to do it with MGI. Here, we will merge all of the data, run it through multiple analysis pipelines, generate Oxford Nanopore data on top of it, and build a gold set that we will then work with. This data will be QC'd, and these QC metrics will be compared across laboratories. In addition, each laboratory can provide a somatic mutation or somatic variant call set, and we will compare that against the gold set. Within this, we will run a second challenge, and that will be that we will give one data set to all analysts who want, make them run it through their somatic mutation calling pipelines, and we will compare that with the gold set as well, and then produce 2 performance reports, one for the entire pipeline, basically going from sequencing through somatic mutation calling, or from data to somatic mutation calling. Now, it's been a long time in preparation, and we've built task forces that have worked on very different ones of the aspects that go together with this. And here you see the entities that are helping with this, very close to Heidelberg, DKFZ, obviously, and all of these laboratories are helping with with how we do the calling, what parameters we use, how we do the pipelining, and so on. This is all a joint piece of work, a huge piece of work. And here, what you can see into what I showed you before, where all of these different task forces have been active. So how do you calculate QC metrics? How much coverage do you need? How do you generate goal sets? So there's a big story behind all of this. And the objective is, in the end, to be able to say, okay, this is how well these laboratories perform, and these are guidelines that allow you to do a good genome analysis. So this is now just about in its starting blocks. Here are some of the QC metrics that we've decided we will focus on, and as I said, this is nearly ready to go. Here, just a couple of the tools that will be run by different ones of the participants. Then all of this used to put it together. This is for small somatic variants, and we have the same thing for large somatic variants. And yeah, so this is a big endeavor, and I hope by the middle of next year we will be able to say, okay, this is a state-of-the-art protocol. These are good thresholds and cutoffs that someone can work by, and these laboratories have this and this level of quality. What I should say is that that in the germline inter-laboratory comparison, we ask the people are on the way to ask for an ISO 17025 or an ISO 15189 accreditation, so that essentially the report can be used by them when they go to their accreditation agency. We have also on the way produced a document that gives a preliminary set of quality metrics for sequencing. It's a living document. It's meant to be vendor agnostic, so it should not be Illumina only. It could be picked up by Illumina or by Oxford Nanopore or by PacBio or by whomever to do their sequencing or to do to sort of follow best practices. But it is a living document that we will refine upon conclusion of the benchmark and the inter-laboratory comparison. So that is how, in the context of a big infrastructure, you can actually work together and move everyone ahead and make sure that they sort of improve technically while they go. The second very important part of the 1+ million genomes and beyond 1 million genomes is this federated, secure, cross-border technical infrastructure. Now, this is a very challenging thing because you need to have some agreement on what you're actually going to put into this system. And very recently, this working group put up a proof of concept. And essentially, they picked rare diseases, very tractable by genome sequencing, and they put up a benchmark. Essentially, they simulated a few genomes. Did I have that on here? No, it's not here. They simulated a few genomes with the viewpoint of two stakeholders in the space. One stakeholder being a researcher clinician, and the second being a data access committee. So how can you cross-border sort of work with each other without actually sharing the data? We generated synthetic genomes. So these were essentially FASTQ files that had variants inside that we peppered into them. I think I even have listed what it is. Yes. So we generated trios. We put pathogenic variants into this RY-R1 gene, and then we put it together with a plausible phenotype. So we put clinical features that go together with this particular variant, and then so we put the questions whether there are the question is, you are working on one system, you've identified this RY-R1 variant, and you want to know whether anyone else on the system has a patient who has a variant in this gene, potentially pathogenic variant, with a similar phenotype. So that is the question, and you want to identify those unfortunate patients. So how did we do this? So essentially, what we did is we built two instances of the RDConnect Genome-Phenome analysis platform. We placed one in Sweden. We placed the other one in Finland. And just to mention, and I will say more about the RTConnect Genome-Phenome analysis platform. This was developed here at SNAG, and yeah, I will explain how this works in a minute. So we put 3 different trios in. These are the phenotypes that are underlying those 3 trios in the Swedish system. These are the phenotypes that underline the 3 trios that are sitting in the Finnish system, and we have together the clinical and phenotypic information keyed up in Ordo HPO and OMI. What I say is the files are FASTQ files, 30-fold coverage genome, and then essentially we produced out of that 25 chromosome G VCFs and put them into the 2 instances. Okay, this is just the functionality standards and rare disease use case that we put in. Key elements inside, obviously you have your data inside, you have a beacon inside, you have Matchmaker Exchange inside. We use all of these different GA4GH standards. We have your credentials basically checked with this Elixir AI. Data sits in here, and then we have the transfer into the Genome-Phenome analysis platform that I will show you later, and then we connect up. Okay, so yeah, Matchmaker Exchange, I will just say that's a connection of different genome phenome computational systems so that you can figure out how the different genotypes or variants that seem causative of disease, how they actually go together with the phenotypes that you are observing. Okay. Again, this is the proof of concept workflow. I will not go into the details of this. What I will show you is here, the federation. So essentially what happened is we had one instance established in in Sweden, another one sitting in Finland. The Swedish instance found this variant, the RY-R1 variant, and then wanted to know, does anyone else have this? And then they essentially, through this federation of these instances, it goes through, it asks for access to the other database, and then can say, tell me with this phenotype and with this genotype pathogenic, do you have someone, yes or no? And then we did this, and it worked. It worked nicely. Okay, in summary, this proof of concept shows that you can put similar databases together and do queries of this type across country borders. The thing that is very important is no data was moved around. So there was no BAM file or no VCF or nothing like that was moved. All that happened was that we had the information structured in the 2 sides in the same way, and we could query from one to the other fairly simply. So it proves the point that you actually can do this, and if you can do it between 2 entities, you can do it between 24 entities. It would not be a huge, it would be a big challenge, but it would not be an unsurmountable challenge. If you want to see a video about this, I want to point you to this thing. Dylan Spalding has generated a video of this, so you can actually go in, click it, and it takes you through, I don't know, a 15-minute or so video where he shows all of the steps that he does. I do this, I do this, I do this, and this is how I finally get my result coming out the back. Okay, which takes me to the last part of what I want to talk about, which is sort of anchored in the International Rare Disease Research Consortium that you've surely all heard about extensively, and it is essentially our RTConnect Genome-Phenome analysis platform. So what is this? The RTConnect Genome-Phenome analysis platform is an online system that facilitates collating, sharing, analyzing, and interpreting the integrated genome-phenome datasets with a particular focus on rare disease diagnosis and gene discovery.
Ivo, make sure
Yes.
you're finished within 5 minutes so that we can have time for discussion.
I will try. Good.
Okay.
So this is what it is. At the moment, there are 600 registers, roughly 20,000 phenome data sets that are present in this system. So just the funding sources that paid for this and the projects that we did to sort of develop this further over time, starting out with RT Connect. On the top are the European projects, on the bottom the national Spanish projects. Okay, so this one I can skip. It just shows you that we've actually federated 3 instances within our own country, so that we can do different instances, all of them talking to each other. So the federation, here I say that this is in planning, but as I showed you before, we did a synthetic example and you can actually see that you can federate. One thing that is important is how you input the phenotype information. So we built a piece of software which is called PhenoStore that allows you with a few clicks to bring in the phenotype of a patient. I will not go into the details of this, but you will understand that having a good ontology is critical, and obviously keying things up into HPO or OMIM and ODO is very relevant. We built disease forms, so we worked with different rare disease ERNs to make specific forms that allow them to input their data very rapidly. So that has happened, and that we can do fast. It aligns with the GA4GH pheno packets standard. This is this means it makes a lot of things interoperable. And this is essentially what happens inside the system. So you have a clinician researcher who has a patient, draws a sample, gives it to a sequencing laboratory, they do their work, generate FASTQs, BAMs, CRAMs, give this to the RT Connect standard analysis pipeline. This can be taken out into EGA, also integrated into the RT Connect Genome-Phenome analysis platform. Clinician inputs the phenotype data using this PhenoStore, and then afterwards gets a connection up to a whole set of analysis tools that allow the researcher to do many different things. For example, set filtering, get annotation of SNVs, indels, copy number variants, can filter by gene of interest, have a phenotype-driven strategy for variant prioritization, link up to ClinVar, look for runs of homozygosity, link out multiple external sources, and use different sources from wherever. This is how it shows itself once you've actually run a query. So you can connect different patients or patient and parents to them and say, I think it's this kind of genetic model, filter it in this way and prioritize the variants for me based on different criteria. You can set this up very fast, and then you get a list of variants ordered from the potentially most pathogenic to the list. So you can have all, or you can have it specific with genes for that particular phenotype. Links, you can generate a clinical report out of that. Comes in PDF format if you want it that way. And then one of the very nice new features that we have on this is this HTS get function that we've linked into it. So you can go in and say, show me what this looks like in the pilot. So show me this variant. I want to see whether I actually trust this or not. So it then goes into the EGA, pulls out the reads and displays them like this. And you can say, yes, I think this is correct, and you can go with that. And then just to close off, so I showed you everything about the rare disease variant of this pipeline. We've also built a cancer variant of this pipeline. And obviously, the easy thing to do is hereditary cancer. So germline variants is very similar or practically the same as the rare disease case. But we also have the somatic mutations inside there. It does the same thing. You run it through the same processes. It has uses DATK best practices for somatic mutation identification. Has a few additional resources in it, the cancer genome interpreter and COSMIC, obviously, and then annotates with that as well. And then you can look at the somatic mutations that are in this particular patient. This is obviously coming out of a tumor normal pair. And I think with that, yeah, you can then pick out the annotation that you like most, see where this comes from, where is it annotated, what is the prediction on it, and then work the way that way. Okay, and with that, I'm going to stop. I'm going to thank these guys here. This is the bioinformatics unit led by Sergi Beltran. They do all of this development of the RD Connect and so on. So I will particularly focus on these guys. Obviously, many other people of the 1+ million genomes who also contribute into this and into the working groups and so on. But I would need 5 slides to put all the names of them of theirs also up here. So with that, I will conclude, and if you have any questions, please let me know.